Okay, I'm going to do a quick video review of the epithelial cell question that we did in class during topic 19. And I'm also going to go over kind of how we did it, and I'm going to use it as a way to go over the type of transporters that we talked about and review quickly concentration gradients. So to begin with this, I want to make sure that we're all on the same page about a couple things, and mainly how to deal with concentration gradients. So very simply, and I'm sure you already know this, but if you have an area here that say is high in concentration of a certain molecule, say molecule A, that molecule, if given the opportunity, will diffuse away from where it's high in concentration to places where it's low in concentration, and it'll do so to a point until it reaches equilibrium where there's equal concentration in the entire area that it occupies. Okay, so this is just simple diffusion. Hopefully a phenomenon that you're very familiar with from chemistry and from general biology days. But let's look at something in a different context. Let's imagine again that you had a very high concentration of molecule A, but in this time you're in a constrained environment such as a tube, a hollow tube. Well, through diffusion, through a spontaneous random effect, molecule A will diffuse down the tube in a fashion so that there'll be less molecules. Oops, that's black, so let's go to red. There'll be maybe a few molecules A down here, a very high amount there, and over time it'll diffuse down until it reaches equilibrium. But before it reaches equilibrium, there is what we call a concentration gradient, because there's very high concentration here, and there's very low concentration here, and in between there's a gradient of mixtures of high and low, essentially. And this is what we refer to as a concentration gradient. And it can be drawn and symbolized by, sometimes you'll see it shown like this, where it's essentially a triangle, where the higher the area of the triangle here, the higher the concentration of the molecule, and the lower the area of the triangle, the lower the concentration of the molecule. Okay, so this is a gradient, and when we talk about molecules moving within a gradient, if, for example, through passive diffusion, the molecules go from a high region of concentration to a low region of concentration, we call this going down the concentration gradient, or it's also been called going with the concentration gradient, because this is a passive event and it's very favorable to happen randomly. Okay, this is favorable because this would just happen by diffusion. If, for instance, though, let's go back here and say I want to take this molecule and I want to move it back up to the region where it's very high in concentration. Okay, so if we're going to move in this direction, we call this going up the gradient or against the concentration gradient. Okay, and this is unfavorable, and this typically will require energy. All right, so down the concentration gradient with the concentration gradient is a passive process. It happens through diffusion. Up the concentration gradient or against the concentration gradient here is an active process, because we have to physically take this somehow and move it where it doesn't want to go. Okay, so understand the terminology of concentration gradients, because it will help you when dealing with the epithelial cell question that we did in class. So here's the epithelial cell example that I gave you in class. And I told you that you were under starvation conditions in this example. Now let's get a couple things straight first of all. This is an intestinal epithelial cell, which means that an intestine, in your intestine is essentially a hollow tube, and that hollow tube is lined with a series of cells called intestinal epithelial cells. Okay, the inside of the tube here is called the lumen, and outside of the tube, right, this area down here is essentially where your bloodstream is and a lot of extracellular matrix protein. So this is the extracellular environment on the outside. Okay, so if you go over this example, intestinal epithelial cells essentially point towards the lumen. So your lumen is up here, and your bloodstream is down here. Okay, and what they do is they use these finger-like projections here And if you remember the term I use, these are called microvilli. Okay, and what they do is they absorb nutrients and transport them through the cell into your bloodstream. Okay, so anything you eat can be digested, pass through your intestine, and the nutrients are absorbed through these cells into your bloodstream. So that's kind of the geography that we're going to be dealing with. Now, I told you that you were under starvation conditions, and we're going to deal with how glucose is taken in from cells, from the lumen of the intestine into cells. So under starvation conditions, I told you that glucose is very low in the lumen because you haven't eaten anything, so it's very low amounts of glucose, and your bloodstream also has very low amounts of glucose. I also told you that during starvation conditions, through a process called gluconeogenesis, the cell makes its own glucose, okay? And that's how it combats starvation. So what you should do whenever I give you these scenarios is immediately write, and with respect to glucose, where the concentrations are. So in here, you have high amounts of glucose in the cell through gluconeogenesis. In the lumen, you have low glucose. And in the bloodstream, you have low glucose. So again, if you think about these as concentration gradients, you can draw these triangles that we refer to. Okay, and here are your concentration gradients for glucose with respect to the inside of the cell, the lumen, and the bloodstream. The other thing, ions that we're going to be dealing with are sodium and potassium. And just remember that there's always going to be very high sodium on the outside of cells, low sodium on the inside of cells. Okay, so again, down by the bloodstream, there'll be high sodium. And then the converse with potassium. There's going to be low potassium on the outside of the cells, high potassium on the inside of cells, low potassium on the outside of cells. Okay, so again, just keep in mind all of your concentrations of these molecules that we're going to deal with in the example. So under starvation conditions, this is kind of how you would have your molecules, or these would be the concentration gradients that we'd be dealing with. What you should also practice doing is thinking about what happens if you had a very rich meal in glucose. What would these concentration gradients be? Okay, so let's start with a blank example of the epithelial cell I gave you. And I asked you to do a variety of things. I asked you to label and diagram different transporters and components of the cell. The first thing I asked you to do was to label the apical side and the basolateral side of the epithelial cells. So just remember that your microvilli point to the lumen of the intestine and that this is the apical side, or the top of the cell. And then the basolateral side are the side of the cells. Okay, basolateral side are the side of the cells that point towards the bloodstream. All right, so there are your apical and basolateral sides, and we can write lumen up here if you want. Again, just to remind us that that's where the lumen is. The other thing I asked you to draw was where are the tight junctions. Now remember that tight junctions are these protein, transmembrane protein pieces that essentially link two cells to each other. Okay, so they're a form of cell-to-cell linkages. But they also do a variety of other things. One thing they do is they prevent all of the fluid that's in the lumen from getting in between the cells and going into the bloodstream. So there's lots of nasty enzymes or digestive things in the lumen of your intestine, and you want to prevent them from leaking between cells and getting into your bloodstream because you don't want those nasty enzymes in your bloodstream. So that's what the tight junctions do. They essentially prevent liquid from flowing in, so the liquid can get into here, but it will stop right here, okay? The other thing that tight junctions do is that they polarize the cell. Remember, a polarized cell is a cell that has a different top than a bottom, and so the tight junctions prevent membrane proteins that maybe are in this side of the cell from diffusing up into the apical side of the cell. All right, and as we do this example, you'll see why you want to prevent that from happening. So tight junctions prevent fluid from coming into the bloodstream from the gut, and it also helps polarize the cell so that cells can have an apical side and cells can also have a basolateral side. All right, so let's do the transporters that I wanted you to have labeled, and we're going to do them one by one and take them step by step. So the first transporter I wanted you to draw was the sodium glucose symporter. Okay, that transporter is found on the apical membrane of the cell, and you can draw it anywhere you want inside the membrane. I'm just going to draw it in between the microvilli, but in reality, these are throughout all of the apical membrane up here. Okay, there's hundreds of them. All right, so let me find a way to label this. So let's go over here. Okay, so this is the sodium glucose symporter. All right, and I'm going to change colors real quick because green probably isn't the best color. Let's just go to black. And remember, a couple things about this symporter. It's a form of coupled transport. Okay, so let's go up here and write some facts about this. Coupled transport is also a form of active transport. Okay, this immediately tells you that you're going to be moving something against a concentration gradient. Symporter means both molecules moves in the same direction. Okay, same direction. The opposite to that is an antiporter, where a molecule moves in opposite directions. And coupled transport is active because the energy comes from moving one molecule down a concentration gradient, and that energy provides the energy to move a molecule up a concentration gradient. So something is going down, and at the same time some other molecule is going up, and that's coupled transport, a form of active transport. Okay, so if you know that, you know the two molecules being moved are sodium and glucose. All right, and they're both being moved in the same direction because it's a symporter. So that means that sodium and glucose from the outside of the cell are being moved to the inside of the cell. Okay, so what I want you to know is the direction these molecules are moving. And then I also want you to tell me for each of these molecules whether it's going up or down its concentration gradient. So if you've already have this part drawn, you're already there. Now if we go over to our other image that we showed, remember, we showed that there is low glucose on the outside of the cell and high glucose on the inside. And so what that means is that glucose here is going from low to high, which means that's going up the concentration gradient or against the gradient. Over here you also wrote that there was high sodium on the outside of the cell and low sodium on the inside. So if you go over here, that means sodium is going from high to low, which means it's going down its concentration gradient. And you've completed the symporter here. And so you can see here, sodium moving down its concentration gradient provides the energy to move glucose in the same direction, i.e. a symporter, up its concentration gradient. And that's the sodium glucose symporter. Okay? The next transporter I wanted you to draw was the glucose uniporter. Right, so let's just draw a block here. Now the glucose uniporter is in the basolateral membrane of the cell. Okay, and I'll explain to you why it's located down here in a second. But remember, this is the glucose. You can call it transporter. I call it uniporter because it tells you that it does only one form of transport. And remember, this transports glucose in one direction. It's passive, meaning the direction is dependent upon the concentration gradient of glucose. Okay, so let's go back and remember, where is glucose high in the cell? Glucose is high on the inside of the cell and low in the bloodstream under starvation conditions. So therefore, glucose will move down its concentration gradient through the uniporter into the bloodstream. So this is going down, concentration gradient. Okay, and that's all that the glucose uniporter does. All right, and then the last transporter I wanted you to draw was the sodium-potassium ATPase pump. Now this pump is actually ubiquitous in all cell membranes, which means it's all over the cell. It's not relegated to the apical or the basolateral side of the cell. So you can draw it anywhere you want. I'm going to put it down here in the basolateral side just so we have some space to work with it. Okay, so let's label this as well. This is I'm going to label it from over here. This is the sodium, potassium, ATPase pump. Now, ATPase pump means it's active transport, okay, which means it's moving some molecule against its concentration gradient, and the energy coming from that is from the hydrolysis of ATP. And so what it does is it actually moves both sodium and potassium against their concentration gradients. So if you go back to our cell gradients, you made notes on that there's high sodium on the outside and low on the inside, and there's high potassium on the inside and low potassium on the outside. Okay? So let's go back real quick. So if you're moving both molecules against their gradients, that means you're going to move potassium from the outside of the cell where it's low to the inside of the cell where it's high. So let's go ahead and draw that. Okay, so potassium from the outside of the cell where it's low, let's just draw low, and that moves inside the cell to where there's high potassium against the concentration gradient. So this is going up. You're also moving sodium up a concentration gradient from the inside of the cell to the outside of the cell. Okay? And this also is up its concentration gradient. So the ATPase pump is moving both sodium and potassium against their concentration gradients using the energy of ATPase to do this. All right, so let's look at how all of this works and together. Remember, we're under starvation conditions, your cell is going to start making glucose through gluconeogenesis, but you also want to maximize taking up as much glucose from your intestines as possible. So the way your cell does this is it uses the sodium gradient. Remember, there's always high sodium on the outside, low on the inside. It uses that energy to couple the transport of glucose into the cell. Okay? Now that we have glucose in the cell, we want to get it into the bloodstream so it can be delivered to other cell types. The glucose uniporter is in the basolateral membrane and now uses the existing glucose concentration gradient because it's high inside the cell and low outside the cell, and through passive transport transports the glucose into the bloodstream. This also shows you why the tight junctions are so important, because the tight junctions prevent these transmembrane transporters to diffuse and move into the apical side of the cell. Let's take the glucose uniporter. If you put the uniporter on the apical side of the cell, what will happen is as you take in glucose from your intestine, that glucose will essentially just be lost by being pumped right back out or being transported right back out into the intestine, and you wouldn't be able to absorb that glucose, you would excrete it. So by making the uniporter in the basolateral side of the membrane, you can make sure that the glucose you're taking in from the intestine is being transported into the bloodstream. The sodium-potassium ATPase pump is in every single cell. All cells have these pumps because essentially they're constantly setting up two large concentration gradients. And we talked about them over here. The sodium-potassium ATPase pump is responsible for making sure that the inside of the cell has high potassium and the outside of the cell has high sodium. All right? And again, this example, we're utilizing the high sodium concentration gradient to provide the energy to bring in glucose. So all three of these transporters are kind of working together to make sure that you maximize glucose import into the cell and glucose delivery to the bloodstream. Now, the last question I asked you was what would happen to glucose, what would happen to the import of glucose if the sodium-potassium ATPase pump was broken? So let's go here and just show right here that this does not work anymore. This is broken. What would happen to glucose import after that effect? Well, what happens is you're no longer pumping sodium out of the cell because this pump is broken. So as you're bringing in sodium down its concentration gradient, the sodium is building up in the cell. So now let's draw a bunch of sodiums here. Okay, you're getting lots and lots of sodiums inside the cell, and they can't be transported back out of the cell. Let's go over here and look at our concentration gradients. What happens is, what's supposed to happen is there's supposed to be a high amount of sodium on the outside and a low amount of sodium on the inside. Well, if the sodium builds up on the inside, this will now turn into a high concentration of sodium, and you'll basically have equal concentrations of sodium on the outside of the cell and the inside of the cell, and there will no longer be a sodium concentration gradient. Okay, so if the sodium-potassium ATPase pump breaks, you no longer have a sodium gradient anymore. If you no longer have a sodium gradient, this sodium can't move down a concentration gradient through the symporter, which means there's no more energy left to couple that movement to glucose import, because there's no energy to take glucose and move it up its concentration gradient if the sodium concentration gradient doesn't exist anymore. So therefore, if the sodium-potassium ATPase pump is broken, you lose the sodium concentration gradient, and therefore you'll lose the ability for the sodium-glucose symporter to import glucose into the cell, and now the cell will not be able to import glucose anymore, and you won't be able to maximize uptake of glucose. Okay, so if you have any questions on that problem, please let me know. It's very important to understand that, one, obviously you're not going to import glucose anymore, but I want you to know the reason why. It's because we're getting rid of the sodium concentration gradient, which is set up by the sodium-potassium ATPase pump. These set up the concentration gradients of sodium and potassium. So overall, that's the epithelial cell problem. I want you to focus on several things. I want you to focus on the location of these transporters. I want you to focus on the nomenclature of what's the apical and the basolateral side of the cells. I want you to know which molecules these transporters actually move and in which direction, and then also whether or not they're going up or down a concentration gradient. And one thing you can do to practice this is, I told you that this example is for starvation scenario, okay, where low glucose in the lumen, high in the cell. Well, you can maybe draw this and reverse the scenario. Do it where you've just eaten a very rich meal. Okay, so now everything's changed. You have very high glucose outside the cell and low glucose on the inside of the cell. Okay, and kind of ask yourself what would happen in that scenario. So just practice getting used to all of these different terms about concentration gradients. Know how these transporters work. Go through that topic and understand the mechanism for how they transport these molecules. Again, if you have any questions on this, please feel free to email me and let me know. And I hope this helped you out.